Hello and welcome to my YouTube channel. Today our topic is on bacterial plasma membrane transport mechanisms. Bacteria use several different transport mechanisms: passive diffusion, facilitated diffusion, primary active transport, secondary active transport, and group translocation. Bacteria are able to transport nutrients into the cell even when the concentration of a nutrient inside the cell is higher than the concentration outside. Thus, they are able to move nutrients up a concentration gradient. This is important because bacteria often live in nutrient-poor habitats. Firstly, here we will discuss about passive diffusion. Passive diffusion, often called diffusion or simple diffusion, is the process by which molecules move from a region of higher concentration to one of lower concentration. That is, the molecules move down the concentration gradient. The rate of passive diffusion depends on the size of the concentration gradient between a cell's exterior and its interior. Water and some gases, including O2 and CO2, easily cross the plasma membrane by passive diffusion. During facilitated diffusion, substances move across the plasma membrane with the assistance of transport proteins that are either channels or carriers. This diagram shows the model of facilitated diffusion. You can see in left side it is outside the cell. The solute molecule binds to the outside of the carrier. Then it changes conformation and releases the molecule on the cell interior. The carrier subsequently changes back to its original shape and is ready to pick up another molecule. Firstly, we should know about active transport. Active transport is the transport of solute molecules to higher concentrations with the input of metabolic energy. Active transport resembles facilitated diffusion in that it involves carrier proteins. Active transport differs from facilitated diffusion because it uses metabolic energy and can concentrate substances within the cell. Three types of active transport are observed in bacteria: primary active transport, secondary active transport, and group translocation. Primary active transport is mediated by carriers called primary active transporters. They use energy provided by ATP hydrolysis to move substances against a concentration gradient. Carrier proteins can be uniporters or cotransporters. You can see in diagram that uniporters move a single substance into the cell, while cotransporters simultaneously move two substances across the membrane. When both substances move in the same direction, the carrier is a symporter. When the two substances move in opposite directions, the carrier is an antiporter. ATP-binding cassette transporters, also known as ABC transporters, are important primary active transporters that are used for import and export of substances, in particular proteins. In this diagram, you can see that ABC transporters consist of two hydrophobic membrane-spanning regions or domains with two ATP-binding domains facing the cytoplasm. A transporter that works with a solute-binding protein free in the periplasm. After binding solute, the solute-binding protein approaches ABC transporter. Solute-binding protein attaches to transporter and releases solute. Energy released by hydrolysis of ATP drives movement of solute across membrane. Secondary active transport couples the potential energy of ion gradients to transport of substances without modifying them. Secondary active transporters are cotransporters. They move to substances simultaneously. The distinguishing characteristic of group translocation is that a molecule is chemically modified as it is brought into the cell. The best-known group translocation system is the phosphoenolpyruvate sugar phosphotransferase system (PTS). The PTS transports a variety of sugars while phosphorylating them, using phosphoenolpyruvate as the phosphate donor. Phosphoenolpyruvate is a high-energy molecule that can be used to synthesize ATP. If you like this video, then please hit the like and subscribe button. Thank you.